Oh yeah, back again. This is the second part of the meiosis sort of explanations. And we're starting to look now at the importance of meiosis. And specifically trying to answer this question, how does meiosis lead to increased genetic variation? And there are two processes that take place, or two events that take place during meiosis that can increase the genetic variation. Now, chronologically, the first of those is a process called crossing over. Now, in the notes, crossing over actually comes after the other event, which is independent segregation. I'm doing it chronologically, so in order of this taking place. The only reason in the notes it comes second is it doesn't happen with every meiotic division, with every cell division by meiosis. But it is the first one that happens if it were going to. So, crossing over takes place during Prophase One. So Prophase One, the very first stage of Meiosis 1. We can see during Prophase 1, we know those events from thinking back to meiosis. So the chromosomes appear as these pairs of sister chromatids. Just remind ourselves to explain that appearance. They are one pair of sister chromatids joined by a centromere, and they're the product of DNA replication. So they exist here as pairs of sister chromatids held by the centromere. They are visible under a light microscope or an optical microscope because they have become condensed. Now, at this stage, in Prophase 1 of meiosis, an event takes place which is different. It's one of the differences to mitosis, which is the formation of what are called bivalents. So this diagram here shows a bivalent. So a bivalent is two of the homologous chromosomes lined up perfectly alongside each other. Now this doesn't take place in mitosis. The homologous chromosomes do not pair up. So a bivalent is where those homologous pairs actually pair up together to form this bivalent, and we can see those bivalents alongside each other during Prophase 1 of meiosis. And that always happens. Now, as they do that, sometimes they tangle up, they wrap around each other. So the sister chromatids may overlap from the, let's call this the maternal and the paternal chromatids in this bivalent may overlap. If they do overlap, that point that they overlap, okay, so the point where they physically cross over, this is known as a chiasma. And in fact, if there are more than one point where they cross over, and that can happen, so the plural of chiasma is chiasmata. Now I'm just showing a single point where we've got this crossing over, that's where the name comes from, between our pairs of homologous chromosomes after that bivalent has formed. Now when they cross over in such a way that they are a very, very precise way, it's possible that the strands of DNA, the molecules of DNA, become broken and then recombine with a chromatid from the other member of the homologous pair. And we can see that down here. So crossing over has taken place at the chiasmata. After that, a section of DNA has been exchanged between two sister chromatids in the same homologous pair. So what we can say here is that you could describe it as a length of DNA, section of DNA has been exchanged. I like to use the phrase during crossing over. Now this is the phrase that I always use. I say that maternal and paternal alleles, I'll underline alleles and come back to that in a second, are exchanged. Now the reason I say alleles is because we know that these are homologous chromosomes, homologous pairs, Homologous pairs, we know two chromosomes that have the same genes, but we also know those genes might not all be the same, they may have different alleles. So here, when I describe crossing over, I say that the maternal and paternal alleles are exchanged. That forms new recombinants So a new recombinant is this, are the chromosomes, or I guess the chromatids that have had those exchanged alleles. Now, after meiosis has completed, the gametes may end up with these recombinant chromosomes present in them. Those recombinant chromosomes are a mixture of maternal and paternal alleles, and therefore are different to any of the cells that have existed before. And that answers that question: how does meiosis lead to genetic variation? One of those ways is through crossing over. Is there anything I can add to that? Let me just check. So yeah, I think probably we'll just finish off just saying that this leads to the production of different combinations of alleles in the gametes that are produced at the end of meiosis. Okay, so that's crossing over. We'll come back to the other reason for the increase in genetic variation in a moment, which is independent segregation. Thanks.